A lot this season. In October, we hosted a Dark Matter Day trivia night with Lux Zeppelin researchers around the world. In December, we learned that humans first proved how the sun shines with an experiment that was operating a mile beneath our feet with Princeton cosmologist Dr. Netta Bacall. In January, we watched Science Friction, an award-winning documentary, and we spoke with its creator and producer, Brian Dunning. In February, we learned about the mysteries of neutrinos with the Majorana collaboration, and we celebrated that collaboration's final results, which were 6 years in the making. In March, we saw how Caterpillar is recreating GPS deep underground. In April, SURF Executive Director Mike Headley gave a laboratory update, and we learned how surf crews provide fresh air throughout the underground. And tonight, we are peering into the gut microbiomes of a honeybee. So this incredible lineup would not have been possible without our Deep Talk sponsors. We want to thank Crow Peak Brewing Company, RCS Construction, Northern Hills Federal Credit Union, Edward Jones, and Chuck and Jolene Lichten-Wallner. Can we give these groups a round of applause? Thank you. If you'd like to support this event or any of SURF's education and public outreach activities, you can do so by getting in touch with SURF Foundation Director Michelle Kane. We have a great presentation for you tonight. Yep, there's Michelle right over there. Dr. Kayla Miller is an assistant professor of biology at Dakota Wesleyan University. She studies the relationship between microbiomes and their hosts, which can be humans, plants, or animals. And tonight, Kayla will discuss how something as tiny as a honeybee gut microbiome could impact South Dakota's entire ecosystem. Please welcome her up.
Thank you so much, Erin, for the introduction, and thanks to all of you guys for coming out tonight. I'm surprised that you get the beer first and then actually stick around for this part. Usually they leave like the carrot at the end to make sure you stay for that, so I appreciate it. If you were trying to come to some of the earlier talks, I'm glad that I did not bring a snowstorm with me this time. So thanks for holding out. So as Erin said, I'm really interested in how bacteria, really tiny little critters, can actually interact with lots of different hosts and have an influence on their host health. And I am interested in the honeybee. And I'm sure you guys have heard a little bit about honeybees. So of course, they're really important pollinators. And they actually contribute up to $18 billion for agriculture in the US. So they are a huge component of this agricultural industry. In this figure, you can see a bunch of different crops and how much they are pollinated by honeybees or other pollinators, and then their crop value. So you can see for some of these crops, they're almost 100% pollinated by honeybees, so they're very important. And then a little bit closer to home, South Dakota is commonly one of the top 5 honey-producing states in the nation. So this is a big industry in South Dakota as well. The other thing you've probably heard about honeybees is that their losses and we see these colonies declining. And the main factor behind these losses is unclear. However, there are probably a lot of things that are kind of adding to this. So it might be pesticides, habitat loss, food quality, or even climate change. So there's a lot of things that are probably involved in this problem. But as you probably have guessed, one part of that I'm interested is thinking about how the bacteria and their gut microbiome might be influencing host health. So I've already used the term a couple of times, but microbiome has kind of turned into a little bit of a buzzword, so I want to make sure that we're all kind of on the same page for what that is. And so it's just a community of microorganisms that's living either within a host or in an environment in that type of ecosystem. And a microorganism is just any living thing you cannot see with a naked eye. So this includes bacteria, which is what I research, but also viruses, fungi, some protists as well. And they can be all in the same community that's all the same microbiome. You have a microbiome in you, enjoying that lovely dinner we just had in your gut, and then of course in many different places on your body as well. So microbiomes are very, very common. Like I said, you have some on you. There are some in cattle, insects, even tiny little worms. Also in our soils, our plants, and even the root ecosystem of plants have their own microbiome. And then the same thing in our aquatic systems as well. So these are very common. I can't say that they're in every organism. Of course, biology always has exceptions, but generally most organisms do have some sort of bacteria or virus or fungi that's associated with them. When we're talking about specific animals or organisms or some sort of living creature, that's when we call these hosts. And like I said, I'm really interested in host microbiome interactions. So the interactions between these insects or animals and their bacterial and fungal community inside. So just like I said, microbiomes are common, host microbiome interactions are very common as well. And I'm going to give you a couple of examples of the good side and then the bad side. So the good side are some of these mycorrhizae, and these are actually fungi, and they form a very close relationship with plants. You can see in this picture here, the mycorrhizae are actually these blue things inside the root cells of this actual plant. So they are actually living inside of the plants. So they have a very good, what we call mutualistic relationship. So there is a benefit for the plant, and then of course there's a benefit for the fungi. So the mycorrhizae, the fungi, are helping to scrounge and get nutrients for the plant, including things that are limited, so phosphorus and nitrogen, as well as water. And in return, the plants are actually secreting carbohydrates out into the ground. So plants do photosynthesis, fixing inorganic CO2 into carbohydrates, and then they're using that to feed these fungus that live in the roots. And so this is a very good mutualism. Mycorrhizae get a place to live, they get some food, and in return, they kick back some nutrients for the plant. On the bad side, I'm going to show you an example a little bit closer to home. So this is a human example. We can go into something we call dysbiosis, or kind of a mix-up of your microbiome. And often this happens from treating yourself with antibiotics if you have an infection. Antibiotics are kind of like a grenade. They get in there and they can kill off a lot of the different healthy microbiome in your body. And what gets left can sometimes lead to overgrowth of an infection or a bad microbe. So this is Clostridium difficile. Some of you guys might know this as C. diff. And this is a very hard infection to get rid of. The best way to cure it actually is through a fecal transplant. So if you want to know more about that, I can tell you later, but we'll leave that out for now. And so that dysbiosis can lead to a lot of different disorders and diseases that can have problems for the host. So that's why it's really important to understand these interactions, either to help sustain and support our beneficial host microbiome interactions, or to help prevent or cure or treat some of these dysbioses that can occur. So why do we look at this inside a honeybee, besides them being super cute? So let's look a little bit at their microbiome. The really cool thing about honeybees is that they have a really simple and consistent microbiome. What I mean by simple is that it's dominated by about 10 to 15 different species of bacteria. In contrast, your gut has over 1,000 different species. So it's a lot harder to study our gut microbiome because we have a lot more species. Also, it's consistent. So honeybees eat nectar, honey, and pollen. That's it. They don't decide to get a pizza one night and grab some fast food another night. They're always eating honey, nectar, and pollen. So their microbiome stays the same pretty much all the time. Whereas, obviously we're eating a lot of different things. And then it's also consistent where my hives in Mitchell, Indiana, the UK, Australia, they all have these same dominant species that we see because, again, they're all eating the exact same thing, even though they might be different plants, but it's basically just sugar and pollen. The other great thing is that we can easily grow them in the lab. So they're very easy to work with, and they're easy to grow and have a lot of it in the lab to do experiments on. And you can see some of this bacteria growing on the plates back in the exhibit hall. So that's why it's great to be a microbial ecologist, because I get to go out in the field and work with bees and look at flowers, and then I can go back in the lab and work with these things there. So overall, that's what makes the honeybee gut microbiome a really great system to look at these host microbiome interactions. We have very few members. We can grow them easily in the lab and ask some of these questions. So a couple of the questions that I'm going to go through tonight are just a couple of parts of my research thinking about how can the microbiome actually influence host health. And I'll talk through a little bit about the research that we have with that. And then I'll move out a little bit broader and think about how the environment can actually influence these different interactions. So we're going to start with how the microbiome can influence the host. And before I get into some of our research, I do need to go through just a little bit more of our bee anatomy. So this is a honeybee. They have these really great hairy legs, and that's where they collect the pollen on their pollen baskets. So if you ever have like a mimic bee or a hoverfly and you're trying to figure out if it's a real bee, check for those hairy legs and pollen, because that'll be a bee. And as I mentioned, I'm really interested in the gut microbiome. So these are the 3 sections of the honeybee gut microbiome. They are very creatively named the foregut, the midgut, and the hindgut. But these are really akin to your stomach, your large intestine, your small intestine. And so each one of these different sections has some different roles. So the foregut is what we call their social stomach, and that's what they fill up with honey if they're gonna go forage. And then as they're foraging about, they're eating the honey and then replacing it with nectar that they'll bring back to the hive. If you've ever heard that bees vomit in each other's mouth, that's true, and that's where the sociality comes in. So they're actually sharing some of that food with each other, and of course their bacteria. This is a very transient ecosystem, so a lot of the bacteria are moving through quickly, so there's actually not a lot of bacteria that stay in that foregut all the time. The midgut is where most of our nutrient absorption goes. So the food that they're eating is getting broken down, the nutrients are getting released back to the bee to help for their metabolic processes. And then in the hindgut, this is where we have these hard to break down materials such as pollen husks or fat globules that are going to require a little bit more work to break them down in order to get those nutrients. So each one of these gut compartments are basically little different ecosystems. And so they have their own unique bacterial community. And so sometimes we're looking at these different sections to kind of get a snapshot of what's happening in each different part. And I'll talk about that a little bit more when I get to my results. So how can bacteria, these little tiny unicellular, very simple organisms, actually do anything to impact host health? They can actually make a really big difference. And one way they do this is through a process of bacterial communication. So bacteria are always producing communication molecules, which are shown as these red triangles here. And you can think of them as kind of like hormones. They're always making them and releasing them into the environment. But if there are very few bacteria around, those signals kind of get lost and dissipate throughout the environment. It's not until they reach a really high cell density that these signal molecules build up and they cause changes in their behavior through gene expression. This is a process called quorum sensing, if you've ever heard of that too. So once they have a quorum, they're able to have these changes in their behavior. And some of those changes in their behavior include virulence factors. So these are anything that they're going to use to cause disease or infections in a host. Antibiotic production, biofilm formation, which we'll talk a little bit more about in a second if you don't know what a biofilm is, light production, and then also toxins. And if you think about it, that makes sense. If you are a little tiny bacteria that wants to produce light, if you're all by your lonesome, you're not really going to make much of a light source for anything. But if there's a huge group of you around, you can actually produce a lot of light. And there's a really cool story about that too that I can tell you about later. So for this research project, we had 2 main questions that we started out with. Are there actually bacteria communicating inside the honeybee gut? And if so, are they affecting any behaviors that might go on to affect the host health? And so this work was done with a couple of undergraduate students. Janae Betcher is a student at Augustana University, and she worked with me over the summer through our South Dakota Bryn undergraduate research program. And then Destiny Brockhouse is a student at Dakota Wesleyan University. Actually, not anymore, she just graduated, but she's been working in my lab on some of these projects. So how do we look for these communication molecules? The first thing we do is actually dissect the bee and take out the gut sections. That's actually really easy to do. The reason bees die when they sting you is because you basically disembowel them as they fly away. So the stinger gets stuck in your skin, and then as they fly away, their whole gut system comes out. So when I'm in the lab, you can just grab that stinger, and you can pull the whole gut sections out, and you're good to go. And we add them to what we call a bioreporter. So this is another bacterial strain, not at all from the honeybee, but this is one that actually senses those communication molecules. And its behavior is that once it has those communication molecules, it produces light. So this is really helpful for us in the lab, because then we can measure that light as a proxy for whether or not those communication molecules are there. So Vibrio harvii is the one that produces this light that I kind of showed you and talked about on the previous slide. So I'm just going to orient you to how this data is going to look. On the y-axis there, we have our light concentration. So the higher up on the scale, the more light that's being produced. And again, remember, we're using that light as a proxy for the presence of these communication molecules. Then on our x-axis, we have our different samples. Either all three gut sections mushed up together, our foregut, our midgut, or our hindgut, and then of course our controls at the end here. And I'll start with just adding the controls so you can see what those look like. So our positive control, a lot of light production, looks great. And then our negative control here. Please note the log axis. I know it doesn't look like they're very different, but I promise you they're statistically very different from one another. And the positive is a very high positive mark. Okay, so now I'll show you the data. So we can see with our whole gut, there's a very strong signal. And then our foregut and our midgut have a little bit less of a signal. So 3 stars means it's a lot, 1 star means it's not quite as much. And then we can see that the whole gut is probably driven from this hindgut. So this hindgut has a very strong signal. A lot of communication molecules, and that made sense to us. What's going on in the hindgut? We have those really hard to break down molecules. They're probably working in cooperation to make the right enzymes, break those things down, and be able to utilize them for nutrients. So that's what we saw with just the communication molecules. So that was kind of the first part. Are there even molecules there? Yes, we're good. Let's move on to the next part. So we were interested in looking at biofilm formation. So biofilms are common in guts. You have one in yours. But biofilms are how bacteria can attach to different surfaces. And they live in these multi-species communities. And they produce these kind of sticky materials that help them stick to the surface and survive there. That's why it's really important in your gut. Things are kind of moving through pretty quickly, so that helps them to attach to your intestinal lining and stick around to help out. If you take your tongue and run them over your teeth and it's a little slimy, that is also a biofilm. So those are just a couple of examples. We also know that biofilms are primarily driven by this quorum sensing, this communication, so it's a good place to look. And we do have preliminary data that shows that these signaling molecules are impacting the amount of biofilm that these bacteria create. So that's what this project's kind of moving forward with, looking at how the biofilm production might be affected by these communication molecules, and then of course how that might be impacting host health. So if you have a good biofilm that's helping to break down nutrients, providing some more of those nutrients for the host, it's going to be beneficial. However, if you have an overgrowth of a biofilm, they could be stealing too many nutrients from the host, and that could be detrimental. So those are some of the questions we're kind of moving forward with this part. So going back to our big questions, how does the microbiome influence the host? We have evidence for microbial communication molecules inside the honeybee. And also, we have some preliminary data that shows that they're influencing that biofilm inside the gut. So next, I'm going to move a little bit out and think a little bit more broadly about how the environment and where these honeybees live might impact their microbiome and what's going on in there. So one thing that the honeybees are exposed to are antibiotics. So honeybee hives have been treated with antibiotics since the 1950s. And this was done prophylactically, which means that they had no disease, no infection, nothing wrong with them. They were just doing it as a preventative measure. And that was important because if you did have an infection, this is what you needed to do. You needed to incinerate your hives, kill all your bees, lose all your equipment, and that's a huge economic loss. So they were kind of doing it as a preventative to prevent this from happening. From the 1950s to about 2017, they primarily used tetracycline, which is an antibiotic you're probably familiar with or you've heard of at least. And then in 2017, we started to change the rules and get a little more strict. Now you need a prescription to treat your hives, and they've also moved on to some newer antibiotics. I put tylazine there, which is an example of one of the newer antibiotics, because you'll see it in a little bit when I talk about my research. So the first part of this story was just kind of figuring out how would these antibiotics change our microbiome, both in abundance, how many bacterial species are living there, and also the diversity, so how many different species are living there. And of course, that's going to impact their health and development by changing up that microbiome. This is work I did during my PhD at Indiana University with my advisor, Dr. Newton, and a master's student at the time, Melissa Blunk, and then our colleague and collaborator at Wellesley, Dr. Matala. So, how do we actually determine who is in the microbiome? What we do is we actually extract all of the genetic material from the honeybee gut, so all of their DNA. So again, we dissect and remove the gut, and then we grind it up and take out all the DNA. Then we sequence that. And sequencing DNA just means that we're figuring out the collection of letters in their DNA sequences, and we can take those to identify the different species that are there. In this data set, I'm going to show you the top 14 different species of bacteria, and they're going to make up 98% of the total community. So like I said, there's usually 10 to 15 species that are common. There's another 2% that are maybe just transient and kind of moving through. These all are unpronounceable names that you don't need to worry about. These just different colors are like different species of bacteria. So like a red oak, a white oak, those pin oak, those are all just kind of different species of bacteria. So the colors are going to be what you're looking at when I start showing you the data. So this is how our experiment was set up. We had two sets of hives. One we called our treatment, and that's the one that was treated with tetracycline from day 0 to day 5. And then, of course, we had a set of control hives that had no antibiotics at all. And then we sampled their microbiomes on day 0, day 15, and day 45. So before antibiotic treatment, a few days after antibiotic treatment ended, and then longer to see kind of what the long-term effects might be. And then on the far left there, get my laser. We have 3 different groups of bees. So our larvae are like the little caterpillar stages of a bee, so they look like little worms. And then we have our nurse bees, which are younger bees that haven't left the hive yet. So they're just in the hive and living there. And then the older bees become foragers, and they're the ones that are actually leaving the hive to go out and forage. Hence their name, foragers. And then within each one of these, we have the treatment and then the control. And then again, at the bottom here, you can see all those different colors of the different species. So I'm going to put all the data up here, which is going to be a little overwhelming at first. But that's just because we didn't see any change. And this is surprising to us. So these are all different days. And you can kind of squint and look between the treatment and control. But we did the stats on it. And there's no difference in the community between the treatment, the antibiotic treatment, and the control hives. The only difference was with the larvae, and you can see that they are quite a bit different than the adult bees, and that's been previously shown before, just the larvae have a different microbiome than adult bees. So, this was a surprise, as science often is. Does anybody have a guess on what happened? And the ladies I told earlier, you can't answer. This is my excuse to get water. Tons of antibiotic resistance. That's what happens when you treat hives with antibiotics since the 1950s without any infection or disease at all. So you're just giving them all these antibiotics, and their microbiome has picked up all this antibiotic resistance. So even though they're getting treated with antibiotics, they basically don't care. There's no change. The microbiome community stays the same. So that was kind of the end of that question, right? We're going to say, how does it change? How does it affect the microbiome? But of course, this high amount of resistance kind of put us on a different path. Again, science. So this work was continued on at Dakota Wesleyan with Dakota Wesleyan student Jeff Shook. He was my research student in the summer of 2020. If you guys remember what happened that summer. And then Destiny's been following this up in my lab right now. And to answer these questions, we actually used a technique that's been used in like clinical settings or therapeutic settings, so when you're dealing with patients. And this is to get an idea of how resistant different bacteria are to different antibiotics. And so what you do is you grow these bacteria with different concentration of antibiotics. So each row is a different species of bacteria. So again, like a pin oak, white oak, different types of species of bacteria. And then on this left side here, we have a really high concentration of antibiotic, and then we dilute it all the way down until it's gone to a really low or no concentration of antibiotic at all. And what they're looking for in a clinical lab is this value here, the lowest concentration of the drug they can give the patient to clear the infection, but that it's going to be the least toxic. So that's kind of how this is used in a medical setting. We're just using it to get an idea of how resistant these different bacteria are to antibiotics. So if they're growing all the way out to this left side at a really high concentration of antibiotics, we say that they are resistant to them. The antibiotic is in the system, they don't care, they're continuing to grow, they're completely resistant to it. Whereas on the other side, we say that they are sensitive. So if they are getting killed off very quickly, they're very sensitive to the antibiotic. So like I said, we look at two different antibiotics. Tetracycline is the old one that they've been using in the past, and we had some evidence for resistance for. And then xylazine is the newer one that they started using around 2017. And this is data from 2020, so only been used for about 3 years. So I'm going to show you some of the data from this project. And don't worry about the numbers, but these are the highest concentration is the darkest color, and then the lowest concentration is the lightest color. And you can see the percentage of our isolates that are resistant to tetracycline. The cutoff for where they treat them at is about 200. So you can see that like a little more than half, maybe 60-ish%, are totally resistant for tetracycline. Not totally surprising. Like I said, we'd seen this already before. So this is kind of checking to just see, yeah, they're still resistant to antibiotics. Okay, now I'm going to show you the xylazine. And just kind of keep your eye on this dark, highest concentration color. Spoiler. And this is just in 3 years. Again, that cutoff is right here, around 200 mg per mil is what they treat them with, which is about 80% of these isolates are completely resistant to it. To a relatively new antibiotic that hasn't been on the market very long. So this is something we're really interested in following up on. Other antibiotics that are they becoming resistant to, and then trying to get back into that question and how it might be impacting the community as well. Maybe not impacting it at all. So, how does the environment influence the interactions? Well, it didn't really change the community at all. So that's why I said we kind of had to pivot a little bit. But now we're really interested in how microbial communities are becoming really resistant to these antibiotics. A little bit for the host health, right? I'm interested in host microbiome interactions. But the global problem of antibiotic resistance is important too. And if this resistance is out in the environment, it's not going to be very long until it's in our hospitals as well. So there's kind of a concern that we have with that too. All right. So going back out to my main questions, again, thinking about how microbiomes and bacteria might be influencing the host. They participate in this communication, changing their behaviors, and those changes in behaviors can impact host health. And then also thinking about how the environment can impact the interactions. And like I said, didn't really see anything because they were resistant to all antibiotics, but that's kind of led us down a new avenue to research. So before I finish up, I just wanted to kind of go a little bit broader back out, and sort of a common question I get when I present some of my research is that people are always really concerned how to save the bees, what can we do to save the bees. And I know I talked about it a little at the beginning, we are seeing these colony losses, but the truth is that these bees are gonna be fine. So this is an example of shipping bees across the country. The 80 bee colony from Bruce, South Dakota, ships their bees to California to pollinate almonds. They get loaded up on these huge semis and moved around. These are domesticated animals, even though they sting you, right? So kind of saying that we're worried about honeybees going extinct is saying like maybe cows are going extinct, right? They're gonna be okay. So I stole this figure from Minnesota, but we have the same data here in South Dakota. So honeybees, there's only one species in South Dakota, and it's the European honeybee. It's not even from here. They're not at risk for extinction. They're gonna be fine. And I don't wanna, if you're a honeybee owner and a honey producer, it's still devastating economically, but overall, they're gonna be okay. What we should be more worried about are over 400 species of native bees that live in South Dakota. And these are often dependent on native flowers. Prairies are most endangered ecosystem, and so they're losing the food that they need in order to grow and survive. So North America has over 4,000 native bee species. Some of them are shown here. This guy especially, this is the rusty-patched bumblebee, is on the endangered species list. So some of these are very important that we start protecting, and do so by, one thing we can do is to help plant native plants in order to help support their communities. So some of you have already been back and seen the seeds. The seeds were donated to us from the Native Plant Initiative through South Dakota State and the Extension Office. And so they have been working to kind of increase the amount of native plants that are out in the world so you can plant it in your gardens. And like I said, you have some seeds that you can take home with you today. And with that, I will leave you with my favorite bee GIF, and then I will have time for questions. So please enjoy this GIF of Oprah Winfrey giving away bees. And they're very happy about it. So if you're not familiar with the episode, this is where she gave out cars. So what they're really excited about are cars. And with that, I'm happy to take any questions.
You're not about to release any bees.
No, look at your chairs.
All right, so we are live streaming this, and we want to capture all of your questions on the mic. So if you have a question, raise your hand, and I'll run over to you and let you speak it into the mic. So I'll hold it. You can just talk it.
I'm not sure whether I missed what was the change or the reason for the change in the antibiotic in 2017, and do you think that's kind of driving this change in the loss of bees? I mean, that's it
was a change in the amount of antibiotic resistance. So I kind of didn't tell you that to give away the story. But yeah, they were seeing this high amount of antibiotic resistance that was happening and trying to kind of respond to that. So like I said, you have to get a prescription now to get antibiotics. But if any of you guys have been to Tractor Supply or Runnings, you can just go buy a huge tub of antibiotics. So I'm not sure how much it's really helping, but that was one of the changes that they'd done.
In your experiment where you said there wasn't a difference between the tetracycline group and the control group, if you had found material from a really remote part of the world where there hadn't been the tetracycline for 50 years, do you think you would have seen a difference?
Yes, and I can say that confidently because the research has been done. So they found feral hives that have been out wild, never been commercialized, and in Europe, and they still have some antibiotic resistance. It's much less, and when you look at European hives or other countries that have banned antibiotics for honeybees, their resistance is also less, but it is still present. So antibiotic resistance is a natural thing that bacteria do, so it's just part of their kind of adaptations. So it's not weird that they would have it, but in America where we treat them a lot, there is much more resistance.
The bees, are they more prevalent out in the fields and the prairies or in the Black Hills?
Native bees? Yes. That is a great question. I don't think I have a correct answer for you. I would guess that it would be more the prairies just because there's more flowers and nesting habitats for them, but it's not something I've looked into personally, so I don't want to say that for sure.
I was wondering, we talk about like super bacterias. How come these guys couldn't evolve to become did they evolve to some super bacteria in the guts, or due to the antibiotic treatment?
Can you explain what you mean by super bacteria?
Means these are resistant to bacteria. The antibiotics. So are they became like some kind of genetic changes they made so that they are now resistant to like wide spectrum,
Yeah, wide spectrum antibiotic resistance. Yeah, so they usually pick up these genes on small chromosomes called plasmids, and they are actually really easy for them to move around. That's another reason why bacterial antibiotic resistance is bad, because they can share it with other bacteria really easily. And some of those resistant factors can be similar for multiple antibiotics. Sometimes they're specific to tetracycline or whatever type of antibiotic. However, that resistance comes with a cost. So if some of the ways that they use resistance is either, blocking the transporter, coming up with an alternative enzymatic pathway, making a new enzyme, all of those things have a cost to the bacteria that they wouldn't make unless they needed it. So that's why I think that, we've moved away from tetracycline, so I think we're starting to see those resistance genes go away, and then now maybe they're gathering some new ones. And I'm not quite sure that answered your question fully, but a little bit, I think.
Okay. My question is regarding, obviously you've looked at antibiotics, but bees are exposed to other chemicals too, such as treatments for mites. Is that something else that you guys have been researching?
I personally haven't looked into it. One of the common things is like formic acid that they treat them with. And I'm not sure if anyone's looked at the impact on the microbiome for that. But that's a good question, somewhere to look.
I'm kind of a science neophyte, so when you talk about something creating light, do you mean light like our indoor lights, or what is the light that's being produced?
It's something called bioluminescence, and thank you for triggering the bonus slides. So let me pull it up really quick. So the textbook example is this cute little squid called the Hawaiian bobtail squid. They're about the size of your thumb. So you can see it up here, but they're really super tiny. And this glowing in the image is all bacteria. And they actually have a light organ. So they produce a special organ in their body to help house these bacteria to produce the light for them. And they require those bacteria in order to have that developmental change to produce that. So why would they have light? I mean, it looks cool. It's used as a cloaking mechanism. So if there are predators kind of looking below, when they look up, they kind of see this as moonlight. And they can actually adjust how much bacteria they have in there. They have like a lens to change the amount of light depending on the moon cycle. So very cool story, but that's, why they do produce light. A symbiotic relationship with another organism. Yeah, great question.
A few years ago or longer, we heard about killer bees. Were they in that? Do they really exist, and are they a problem, and are they attacking the natives?
They do really exist. They have been in America in a couple of different locations, but I don't think as widespread as we were worried about. And I wouldn't say they might be competing for resources with natives, but not necessarily. They wouldn't be killing them directly. But again, not something I've looked a ton into, so. That was kind of our other 2020, oh God, killer bees. So we had a virus and now.
Can you talk more a little bit how you do the gene sequencing?
Yeah, so what I do is I extract the DNA and I actually amplify it through a process called PCR. And so in order to send it off for sequencing, you need a lot of the DNA. So basically it's a photocopier for our DNA. And the process we use is the exact same thing that they use inside their cells to make copies of their chromosomes. So we actually have the same enzymes that they use inside their cells to make those copies, just kind of on an industrial scale. And then I ship it off to other people that do it. But I know how the process works. So they have the nucleotides, which are the little building blocks of DNA. And they have a little camera that looks at it. And every time one of those nucleotides are added, a little bit of light gets shown. And each light there's 4 bases in DNA. Each one has a different color. And that was kind of represented in that figure I showed you. So they have this tiny little camera that looks for those different bases that come up, and then that color signal goes to a computer that kind of marks it down. Oh, I saw red. I saw really bright red. That's probably 4 T's. I saw a green. That's an A. And that's how they do the sequencing. That's kind of our 2nd generation sequencing. Our 3rd generation sequencing is even more technical, and they use a pH probe, a very, 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 very tiny one. Now, every time one of these bases is added, there's a hydroxide ion released, which changes the pH ever so slightly. And they use that in order to measure the change in pH to get the DNA sequences. It's a lot faster, it's a lot cheaper, and it's made a lot of this work more accessible in just the last couple of decades. So there was actually just an update to the human genome. They originally had only done 1 person, and that took like 10 years and a lot of work. And now we have the technology. They've actually incorporated a few different samples to have a more broad and diverse human genome. Yeah, good question. And sorry if I lost anyone on nucleotides.
All right, awesome. Thank you so much.
Thank you.
Let's give Kayla a round of applause. All right, so like she said, if you have any other questions, we will be out in the exhibit hall. You can we have some little peat pots that you can do a little seedling starter for some native plants if you want to take some home and start growing them to have some species to help our native bees thrive in South Dakota. We also have some, can you talk a little bit about what they can see in the microscopes out there? Yeah. Oh my God.
So yeah, there are some dissecting microscopes which are a little bit bigger, and there's some honeybees that you can look at a little bit more closely. So they're there in little dishes. Next to the microscopes are just some petri dishes of the bacteria growing. Not super exciting, but if you've never seen it before, it might be interesting. And then also there's a little experiment there on some antibiotic resistance, so you can see a little bit of that too. On the other microscopes are actually microscope slides of some of the bacteria that live inside the honeybee gut, and they're stained so you can see them, and then they're up on the monitors so you can look at them really closely. They're very small, highly magnified for you to be able to see.
Awesome. So definitely stick around and check that out and ask some more questions. Again, thank you guys so much for being here tonight. We will be back in October with another wonderful season of Deep Talks. So we are looking forward to some of the speakers we've already started to line up. If you just can't wait for October, we do have Surf's signature event coming up. Neutrino Day will be on Saturday, July 8th, and that is when we transform the entire city of Lead into a ginormous science festival. We have hoist room tours, we have hands-on activities for all ages, from little kids to adults. We have incredible demonstrations and even some live chats with scientists and researchers who are doing cool things underground. So you get a little window into the underground. And our keynote speaker is Earth's premier science comedian, Brian Mallow, who we had join us in 2020 virtually, and now we get to invite him back in person up at the Opera House. So you can visit neutrinoday.com to see all of the awesome stuff that our team is lining up for Neutrino Day. Thank you again for coming. Have a lovely night. Thanks.